So welcome to the next session, looking at the role of chemistry in drug discovery. The session outcomes of this session are to first understand the role of chemistry within the drug discovery process, to recall how something called CAD, which is known as computer-aided drug design, can be used to generate and modify compounds to create new compounds, and also understand the drug discovery process from drug discovery through to approval. So an introduction. So in the past, most drugs have been discovered either by identifying the active ingredient, and this is from like traditional remedies, so like natural compounds, or by serendipitous discovery. But now we know that diseases are controlled at a molecular and physiological level. Also, the shape of a molecule at an atomic level is well understood. And also we have information now from the Human Genome Project from back in 2001 when it was first published. This is really just showing you like the history of kind of drug discovery. So in the pre-1919, there was like a reliance on kind of herbal drugs and serendipitous discoveries. Around about the 1920s and 1930s, vitamins and vaccines were kind of booming at this point. And then we had the development of the tuberculosis vaccine, and many other vaccines came through. In the 1940s, this was known as the antibiotic era, and there was a big boost to research and development due to World War II. In the 1950s, with the discovery in 1953, discovery of DNA by Watson and Crick, this led to the new technology, so the discovery of DNA. And then in the 1960s, there was kind of a breakthrough in something called etiology. So this is a type of science which looks at the causes and origins. So for example, the cause of high blood pressure is due to smoking, or the lack of exercise or stress, and maybe a diet high in salt and fat. And so this kind of allowed us then to kind of assign a cause to the disease. In the 1970s, we had the rise of the biotechnology, and that was due to the 1950s with the discovery of DNA, and also the use of information technology, IT. In the 1980s, we started to see the commercialization of drug discovery. So many drugs were being patented, and they were coming through for delivery for kind of targeted therapy in cancer. And also the use of combinatorial chemistry, which we'll focus on later. And in the 1990s, we started to use robotics in kind of high throughput screening, and this involved automation of the drug discovery process. So the cost of drug discovery, the cost has actually increased, whilst what we've seen, and we've kind of talked about this before, is that we don't see any increase or big increase in the number of kind of drugs that are coming to market. What we know is that around about 800 million is spent just to bring one new drug to market. So it's a big expenditure. And around about 127 billion is actually spent on pharma and R&D, so research and development, back in 2010. And there's actually a share of something called contract research organisations, and this kind of accounts for 27% of research in the drug discovery market. And the world contract research organisation market is worth around about 16.3 billion. This table kind of summarises some of the top contract research organisations according to kind of the amount of revenue that they generate. So you can see this company here called Quintiles, they generate around about 2.5 billion. We've got Charles River Laboratories. They have a laboratory based over in Discovery Park and also at near Manston Airport. They have a revenue of around about 1.2 billion. And you may recognise some of these companies. Kendall, they have a revenue of around about 590 million. So you can see that some of these contract research organisations, they are generating a lot of revenue due to the sales of the products that they have marketed. Kind of revisiting the kind of drug discovery timeline, so the drug discovery and development. So drug discovery development is kind of a process of bringing the new pharmaceutical drug to the market. That's once you have a lead compound. And that's kind of been identified through the process of drug discovery. So in the beginning, we can be screening around about 10,000 compounds. But in fact, this screen may be much larger, and we could be screening millions of compounds. Then there's a preclinical screen. So we take those 10,000 compounds and then we screen those, and then we select 250 compounds in the preclinical screen. And these two phases, they take around about 6.5 years. Then there's the clinical trial. You may select 1 to 5 compounds to take forward to clinical trial. And this can take a period of 7 years, which will then actually follow into the FDA review and also the marketing. So in all, it can take anything up to 10 to 12 years to bring a drug to market. So what's been evident in the probably the last decade is computer-aided drug design. Computer-aided drug design, also known as CAD, can be used at different stages in drug discovery. So it can be used, for example, at hit identification using virtual screening. So this is in silico screening. And we can look at structure and ligand-based design. And we'll look at this a little bit further on. Also, we can look at hit-to-lead optimization of affinity and selectivity. So this is drug-based design. Also something known as QSAR, quantitative structure-activity relationship. And then after, we can also we can look at lead optimization. So CAD can be also used for lead optimization of other pharmaceutical properties while maintaining affinity. So the use of computer-aided drug design has changed the way that we do drug discovery. In the 1990s, the classical project combination approach would be between chemistry, biology, and drug metabolism, so ADME. ADME, as you remember, stands for absorption, distribution, metabolism, and excretion. And this was it was between these groups. In the after 2000, we take a more automated approach, and we start to then use computational chemistry. So this is CombiChem with high throughput screening, along with ADME. So that's not lost. So these studies then link together in more of a streamlined fashion. So looking at computational aided drug design, so CAD, this is essentially the prediction of binding the affinity. But in order to overcome an insufficient prediction of the binding affinity, this can be calculated by recent scoring functions. The protein-ligand interaction and the compound 3D structure information are used in this analysis. For structure-based drug design, several post-screening analyses focusing on protein interaction or protein-ligand interaction, should we say, has been developed to improve the enrichment and also for mining potential candidates. For structure-based drug design, we can do this through consensus scoring. So this is selecting candidates by voting of multiple scoring functions. And so we may lose the relationship between the protein-ligand structural information and scoring criterion. We also can look at geometric analysis. So this is actually comparing protein-ligand interactions by visually inspecting individual structures, which are becoming intractable when the number of complexes to be analysed is increasing. And cluster analysis, which is quite popular, represent and cluster candidates according to protein-ligand 3D information needs meaningful representation of protein-ligand interactions. Now I'm focusing on the kind of structure-based CAD, known as SB-CAD, and ligand-based CAD. Now, these computational tools are useful as a way to kind of expedite, say, antibiotic drug design process. And these two approaches essentially allow us then to investigate the computer-aided drug design. So for drug-based CAD, we can do things like pharmacomodeling, de novo design, ligand docking, and molecular dynamics. Whilst with ligand-based CAD, we can do pharmacomodeling, QSAR, and virtual screening. Looking at now, these are kind of three methods of drug discovery. In particular, in the preclinical drug discovery, there is a process before the clinical testing of the drugs. And the first steps in the preclinical drug development is the discovery and synthesis of a new drug. So we've got these three methods. We've got chemical modification, we've got rational drug design, and random screening. And these all feed into this drug candidate lead compounds. And also we can also, which is something which is a little bit out from the normal kind of drug discovery method, is serendipity. So serendipity is the discovery of something fortunate, but may also have a role in drug development. Focusing now on the drug discovery methods, it's an important group of drugs which have been made by chemical modification, which are known as diuretics. The initial kind of steps was through serendipity with antibiotic known as sulfonamide, which causes sodium bicarbonate diuresis from the kidneys. Now the drug could be based on this sulfonamide-like action, as this promotes water loss from the body. And so they took the drug, which is known as the structure as chlorophyoside, and they made another drug called furosemide, which is used today in the treatment of hypertension and heart failure to promote water loss. We've also got rational drug design. An example for that here we're going to consider is Sanamiva. The rational drug design, included the kind of the computer-aided design, which is using in silico study. Now, what they showed was that they could prevent and shorten flu infection. And when they kind of studied the mechanism of action, what they found was that the neuroamidase enzyme is involved in viral infection. And in fact, Sansamir binds to this neuraminidase and inhibits the virus. So it actually inhibits, it binds to a pocket within this enzyme and prevents it from replicating. Now focusing on the third approach in the drug discovery, which is random screening. This uses kind of high throughput screening, which is managed by robotics, similar to what we can see in this picture. So there's a random screening for kind of the biological activity, and the screening can be from banks of previously discovered chemical entities, and there's a large number of kind of natural products, as well as peptides and nucleic acids. And random screening has led to the discovery of a drug called cyclosporin, which is immunosuppressant. We're going to focus on different parts of the kind of drug discovery process, but specifically we're going to kind of break this down into the different sections. So first we're going to look at the kind of target selection. So this is things like these are cellular or genetic targets. So these could be proteins, receptors, or DNA. We also can have a look at genomics. So there are kind of masses of data available. For example, from the Broad Institute, where they've analysed the expression of certain genes in cancer cell lines. Proteomic studies, there are a number of papers where they've kind of analysed the secretome, so this means like proteins which are secreted from cancer cells as a way to discover novel biomarkers. And these novel biomarkers may be useful in the therapy route. And also bioinformatics, which actually encompass like genomics and proteomics. The lead optimization, this involves like the synthesis and isolation. And also we used combinatorial chemistry and the assay development. Okay, so the assay development is really important if we want to screen, hundreds of thousands of drugs, which can be done through a high throughput screening. The medicinal chemistry, this is looking at the kind of library development, the SAR studies, the in silico screening, and the chemical synthesis. The in vitro studies, so we use in vitro studies to study things like drug affinity and selectivity. We use the cell disease models, which are specific for the disease that we were targeting, and we look at the mechanism of action. And also then we can then get like a lead candidate refinement. After we have a lead candidate, then we can move into in vivo studies, so using animal models of disease states, and we can perform behavioral studies and also functional imaging. So this is like things like MRI or CT scanning. And then also we can at the same time do ex vivo studies, which is particularly important if we want to study the excretion profile of our drug. And then afterwards we would do clinical trials and therapeutics. Now focusing on target selection. So target selection in drug discovery is defined as the decision to focus on finding an agent with particular biological action that's anticipated to have a therapeutic utility and is influenced by a complex balance of scientific, medical and strategic considerations. Now, to find our target or to identify target, we need to identify the molecular target that are involved in the disease progression. So we need to understand how the disease progresses. And the target validation, this helps us to prove that by manipulating the molecular target, so things through RNAi, so knocking out the target, we can provide a therapeutic benefit for patients. So there are a number of targets that have been looked at previously, and these are kind of the biochemical classes of the current drug targets. Now these are kind of split into different types. These are mainly proteins, including G protein coupled receptors. They occupy the most of the drug targets, around about 45%. We also have enzymes that are around about 28%. Things like hormones and growth factors are around 11%. Ion channels are 5%, and nuclear receptors are the lowest group at 2%. We're going to look at the techniques for target identification. So how do we identify our target? So we can use cellular and genetic targets. So this involves the identification of the function of a potential therapeutic drug target and its role in the disease process. For small molecule drugs, this step in the process involves identification of the target receptor or enzymes, whereas for some biological approaches, the focus is at the gene or transcription level. It depends on the target. Drugs usually act on either a cellular or genetic chemical in the body known as the targets, which is believed to be associated with disease. If we look at the genomics, this is the study of genes and their function. So the genomics aims to understand the structure of the genome, so including the mapping of genes and the sequencing of the DNA. And also it exploits the findings from sequencing of human and other genomes to find new drug targets. And the human genome, that consists of a sequence of around 3 billion nucleotides made up of the nucleotides A, C, G and T, which in turn are probably encode around about 35 to 50,000 genes. The number of genes which are implicated in disease are estimated from a publication from Drew to be around about 1,000 genes which are maybe defected, which may have defects or a misregulation. Now, this is based on 5 or 10 linked genes, linked proteins per gene. And what Drew actually proposed was that there was a number of potential drug targets that may lie between 5,000 and 10,000. Things like single nucleotide polymorphisms, these are known as SNPs, which is pronounced as SNPs. They are the most common type of genetic variation among people. So each SNP represents a difference in a single DNA nucleotide. Okay, and we can compare the genomes from both healthy and sick people to identify where the genomes differ, so where the SNPs are differing. Proteomics. So this is a study of the proteome. So essentially it's the proteins, okay? And there's a kind of complete set of proteins which are produced by species. So these are using kind of technologies of large scale protein separation and identification. So things like mass spectrometry you've probably heard about. We can use this to identify the proteins. It's now becoming increasingly evident that the complexity of a biological system lies at the level of the proteins, and that genomics alone will not suffice to understand these systems. It is also at the protein level that disease processes become manifest, which at most 91% of drugs act. Therefore, the analysis of protein, including protein-protein or interactions or protein nucleic acids and protein ligand interactions, are the most important in target discovery. So proteomics is a kind of systematic way of high throughput separation and characterization of proteins within a biological system. And target identification with proteomics is performed by comparing the protein expression levels in normal and diseased tissues. And things like two-dimensional SDS page is used to separate proteins, which can then be identified and then characterized by liquid chromatography mass spectrometry. So bioinformatics. So bioinformatics essentially is a branch of biology that kind of develops a method and software tools, and it's kind of made up of algorithms and databases, which helps to understand effectively the use of biological data. And it kind of encompasses many parts of science, things like computer science, statistics, biology, mathematics, chemistry, biophysics, engineering, and biochemistry. So we've already said it's a kind of branch of molecular biology. It's a very important, plays an important role in the kind of the drug discovery process. It's particularly important in the kind of target identification and also in the computer screening of chemical compounds and pharmacogenomics. Now, the methods that are used to transform the raw data into this kind of meaningful information, so example from genes and their encoded proteins, and then we can compare to like whole genome, so whether or not there's disease or not. Bioinformatics allows us to make that comparison. So we can compare the entire genome of a pathogenic or non-pathogenic strain of microbes. And we can identify genes and proteins associated with that pathogenism. So using gene expression microarrays, microarrays are essentially, we can take like a microchip, which is then coated and we can use antibodies or we can use nucleic acids. And we can use this, it's called a gene chip technology. It's essentially a single device which can help to evaluate and compare the expression of up to 20,000 genes from healthy and diseased individuals at once. So the lead discovery is kind of the identification of small molecule modulators of protein function and the process of kind of transforming those into a high content series. Now it involves different steps and we'll focus more on these. Focusing now on the synthesis and isolation, so this is the separation of mixtures, separation of impurities, and also the in vitro chemical synthesis and biosynthetic intermediate. Combinatorial chemistry, this is the rapid synthesis of or computer simulation of a large number of different but structurally related molecules. It allows us then to search for new leads, and we can also optimize target affinity and selectivity, and we can also evaluate the ADME properties, as well as reducing toxicity and eliminating side effects. In assay development, we can be used to measure the activity of a drug. We can discriminate between compounds and evaluate whether or not it's going to express protein targets or enzyme substrate interactions in the assay development. And then we can use the assays in the high throughput screening, and that's the screening of the drug target against a selection of chemicals, and that we can then perform the identification of a highly target specific compounds using the automated robotic screens. In medicinal chemistry, this essentially is a discipline at the intersection of the synthetic organic chemistry and pharmacology, and it focuses on small organic molecules and not on the biologics or inorganic compounds, and that's used in drug discovery hits, lead optimization, hit to lead, and process chemistry and development. In the library development, this essentially is the collection of stored chemicals along with the associated database. And actually many pharmaceutical companies have their own databases where they store millions of compounds. And this actually helps or assists in the high throughput screening and helps us then to screen for drug target hits. And this is based on the organic chemistry. If we look at the SARS studies, so this is not COVID, it stands for Structure Activity Relationship. A pharmacophore is kind of a description of a molecular feature that's kind of necessary for molecular recognition of a ligand. So this kind of helps us to identify the pharmacophore. So the pharmacophore is the kind of precise section of a molecule that's responsible for the biological activity, and it enables us to prepare more active compound or allows for the elimination of excessive functionality. If we take, for example, the example of morphine. Morphine is the most abundant of the opiates, of 24 alkaloids, and that's accounting for around 9 to 14% of opium extract by mass. It was named after Roman god of dreams known as Morphus. The drug morphine numbs pain and alters mood and induces sleep. Now the Freezy structure is fascinating. Essentially it consists of 5 rings, of which approximately are in the same plane, and the other 2 rings, including the nitrogen one, are each at right angles to the other. So if we look at the kind of like the biologics action of opioids, this depends on different groups. So for example, we've got the phenolic hydroxyl group, which we find on the aromatic ring. We've also got the 6-hydroxyl group, which we find on the alcoholic ring. And then we have the double bond, which is between this carbon 7 and carbon 8. And then we have the N-methyl group, which is on this, it's called a piperidine ring. And then we have the ether bridge, which is on this kind of tetrahydrofuran ring. And then finally we have the aromatic ring. Now these are known as kind of like functional groups. If we look at the functional groups of morphine, we can actually help to like kind of mask or remove a functional group in the kind of drug discovery process. And we can test the analogue for activity. And this helps us then to determine the importance of, or otherwise, of a functional group for activity. So we can determine whether or not that functional group is required for activity. So these are other examples of opioids. So if we're going to compare morphine with the other groups, with the other compounds. So we've got codeine. So codeine has on this has a methyl group here, and codeine is a methyl morphine, and it occurs naturally in opium, and it's partly converted in the body to morphine. It's less potent than morphine, and it's also it's kind of like a partial agonist to the micro-opioid receptor. Heroin, it's actually around about 3 times more potent than morphine. Is even more lipid soluble, so it can get past the kind of blood-brain barrier, and it has these acetyl groups which can be hydrolyzed. Also, there is this one, which is fentanyl. Fentanyl it's essentially a synthetic opioid, so that's being created in the laboratory. So its structure is kind of really different from the natural or the semi-synthetic opioids. In fact, the core is made up of these. It's a core kind of nitrogen ring, which is still present in the fentanyl, which allows it to be effectively bind to the microreceptor. Moving on now to in silico screening. So in silico screening essentially is computer simulated screening of chemicals, and it helps us to find structures that are most likely to bind to a drug. Okay, so it's kind of it's using computers to do the screening as opposed to a wet bench science, and it helps us to then filter the chemical space. So this can be enormous. Okay. And it's more economical than high throughput screening. However, it doesn't really tell us whether or not the compounds that we get from the screening will work in principle. So the chemical synthesis involves the production of those lead compounds into suitable quantity and quality to allow large-scale animal and eventual extensive human clinical trials, and the optimization of that chemical route for bulk industrial production. And we also need to look at kind of suitable drug formulation, and we'll come to that in the next couple of weeks. Looking at the in vitro studies, so this is essentially means in glass studies, so in the test tube. It's using a component of the organism, but in the test tube. So examples include cells derived from a multicellular organism, so they're grown in kind of tissue culture. We can also perform analysis with subcellular components, things like ribosomes, which make up obviously the protein for protein synthesis, mitochondria, so these are like organelles. We can also look at cellular and subcellular extracts. So in the past, we've looked at things like wheat germ or reticulocyte extract. So we can even make proteins using these, they're essentially like cytosol extracts. And also we can purify molecules, things like DNA and RNA, to perform in vitro studies. So the advantages of in vitro studies are that they can be completed in a very short period of time. It reduces the risk in the kind of post-clinical trials, and it allows us to simplify, well, to enormously simplify the system, and the investor then can focus on a small number of components. Looking at the kind of drug affinity and selectivity. So drug affinity is the ability of a drug to bind to its biological target, so things like receptors, enzymes, and transport system. The selectivity, this is a drug should bind to a specific receptor on the site of a cell, for example, aspirin. So focusing now on the drug affinity. So the drug affinity can change according to the drug, and it depends on the dosage. There's lots of things that will kind of determine how, what is the affinity of a drug. So it might be that it could be something to do with the active site. And if we look at this this graph, the more potent the drug is, it has normally will have a higher affinity. The line will move to the left. So in the line of A, which is the red line here, that's the most it kind of has a high affinity, whilst the lower affinity drug would be this green one here. And what this means is it would take more drug to get something called the 50% dose. Regarding the aspirin, so aspirin is kind of widely used as a non-steroidal steroidal anti-inflammatory drug known as NSAIDs. It has been well documented that aspirin actually irreversibly inhibits cyclooxygenase COX by acetylation, and so it kind of blocks this receptor from working, and then subsequently blocks subsequent biosynthesis of prostaglandins and thromboxane. Focusing on now cell disease models. So these we have different types of models, and here are some examples. So isogenic human disease models. These are a family of cells that are selected or engineered to accurately model the genetics of a specific patient population, and this is done in vitro. Now, isogenic also means that they have the same genetic background. Then stem cell research models. These are from adult or embryonic stem cells. These are carrying or induced to carry defective genes that can be investigated in vitro to understand latent molecular mechanisms and disease characteristics. So how do we refine our lead candidate? We have to optimize the chemical hits for the clinical trial. So this is commonly referred as the lead optimization. And the refinement of the structure is necessary in order to improve, for example, the potency, how effective the drug's going to be at a low dosage, the oral availability. So most drugs are made as capsules for oral route, selectivity, the pharmacokinetic properties, so things like its excretion profile, whether or not it's stable, and then also the safety as well as the ADME properties. When we've got our lead optimization, we have to analyse our drug in vivo studies before moving into clinical trial. And this is essentially the experimentation using a whole or living organism. This provides us with information around the metabolic profile, the toxicology, so how safe our drug, and also the drug-drug interactions. The animal models of disease states. So this tests conditions involved induced disease or injury similar to the human condition. This must be equivalent in mechanism, of course. So we can predict the human toxicity in around about 71% of cases. So for example, we can use something called skid mice for development of HIV treatment. SCID mice stands for severe combined immunodeficiency. So this is what the SCID mice look like. They're kind of naked, but they have a deficient immune system. We also have the node mice. So here's an example of a node mouse. They've been used in diabetes. And the zebrafish, known as danio, they're used for kind of looking at gene function. Regarding behavioural studies, these are tools to investigate behavioural results of drugs. We use them to observe depression and mental disorders. However, self-esteem and suicide are really hard to induce in animals. So examples of behavioural studies include things called despair-based, which is kind of, it's quite distressing to see this actually. It's forced swimming, so the mice are kind of forced to swim, or they're suspended in water. They look at kind of tail suspension. There's also tests where they look at reward-based, so they use food to reward the animals, and also anxiety-based. The functional imaging, this is looking at the method of detecting and measuring changes in the metabolism, so blood flow and regional chemical composition, as well as absorption. So here they can use tracers and probes to track the drug in the body. So normally this would be done first with animal studies. And they can use devices such as MRI scanning or CT scanning. Ex vivo studies, these are looking at the experimentation on tissue in an artificial environment outside the organism with a minimum alteration of natural conditions. There is some kind of ethical issues of using like human tissue in these types of studies. Examples include measuring tissue properties as well as realistic models for surgery. But ex vivo studies are particularly useful if we're studying things like blood-brain barrier, or we're looking at excretion. Regarding the clinical trials, so this is essentially a set of procedures in kind of medical research and drug development. And this allows us to study the safety and efficacy of new drugs. And it's essential to get marketing approval from regulatory authorities. Now, this may take up to 7 years in the process. So phase 1, this essentially is the kind of most recent designation, and it's known as a human microdosing study. So this is kind of really like the first part. So the first is the human trials is conducted to study exploratory or investigational new drugs. So this is designed to speed up the development of promising drugs. And it's concerned with kind of looking at preliminary data on the drugs' pharmacodynamics and pharmacokinetics, and the efficacy of pre-clinical studies. Phase 1, this is the clinical pharmacological evaluation. So this is the first stage of testing in human subjects. And normally they recruit around about 20 to 50 healthy volunteers. And this concerns in this part of the phase, they'll be looking at things like human toxicity, tolerated dose range, pharmacology, and dynamics. So types of phase 1 trials, so things like they'll be doing like single ascending dose, so they'll look at different dosages, multiple ascending dose, and also looking at any interactions with food, so the effect of food in the phase 1. In phase 2, so this is a controlled clinical evaluation, and the patients are enrolled, and they normally look at a bigger cohort of patients, normally between 50 and 300 patients. This is normally controlled as a single blind technique, so meaning that the patients don't know if they're taking the drug or not, so they might be taking a placebo. And in this phase, they want to look at things like safety, efficacy, drug toxicity, and drug interaction. In the next phase, which is phase 3, essentially this is an extended clinical trial, and it's the most expensive and time consuming, and there are around about, it's a bigger cohort, between 250 and 1,000 patients. It's a controlled double-blind technique, and in this phase they're looking at things like safety, efficacy, and then they make a comparison with other drugs. They decide on the development of the packaging of the drug. The phase 4, this is really is the quickest of the phase, and probably not as the most the least expensive. It's the kind of post-marketing surveillance. So they're designed to kind of detect any rare or long-term adverse effects, and they look at adverse drug reaction monitoring and pharmacovigilance. Just going to kind of focus on a couple of techniques now. So we've got gene therapy. So gene therapy, this is a technique which is used for correcting defective genes. And it's a kind of a process to insert genes into the cell to treat disease. And you can use viruses, so such as the adenovector or adenovirus, to correct the defective genes in the target cell. Now, gene therapy is used to kind of correct this deficient phenotype. Now, there are a number of gene therapy approaches. So we have germline gene therapy. So this is where they use sperm or eggs, which are modified by the introduction of functional genes, which are then integrated into their genomes. And so what happens here is they would change the heritable and would be passed on to later generation. So stomatic gene therapy, this is the therapeutic genes which are transferred into the stomatic cells of a patient, and the change will not be inherited by the patient's offspring or later generations. Now there's kind of different types of gene therapy. We've got ex vivo gene therapy. This is the transfer of therapeutic genes in culture cells. So the cells have been taken from the patient, and then they've used the virus to infect the cells. And that's then reintroduced back into the patient. And this was used for therapy of ADA deficiency. In vivo gene therapy, so this is the direct delivery of genes into cells of a particular tissue, and it's referred as the in vivo gene therapy. An example is that therapy for cystic fibrosis. And there's been developments of a nasal spray which can help to deliver therapy for cystic fibrosis. So the gene therapy vectors that are used are viruses, so things like retroviruses, adenoviruses, adeno-associated viruses, or herpes simplex virus. Now the virus is modified so that it can't form an active virus, so it only affects once. But they can also look at using for gene therapy is pure DNA constructs, or things like lipoplexes, also DNA molecular conjugates, and human artificial chromosomes. Anyway, that finishes our session for today. I have the last slide to present to you. This is from further reading. Please take a look at this book chapter from Benjamin Blass in Basic Principles of Drug Discovery and Development. Have a look at the drug discovery process. It's a really nice chapter. Chapter 2, pages 35 to 66. Now this is available as an ebook from Canterbury Christ Church Library. Also, this publication, which I'll upload onto the Blackboard, is looking at the principles of early drug discovery.